Hello, everyone. It's a pleasure to be here on behalf of the PGC TSOCD Working Group. I'm going to be presenting on what is OCD. My name is Dr. Paul Arnold. I'm professor and director at the Mattheisen Center for Mental Health Research and Education in Calgary, Alberta, Canada. We diagnose OCD based on a standardized diagnostic manual known as the DSM-5, and OCD occurs in the presence of either obsessions, compulsions, or both. Obsessions are defined as recurrent intrusive thoughts, urges, or images that are experienced as intrusive and unwanted and that cause marked anxiety or distress. Classic example would be an intrusive thought about excessive contamination in one's environment. They are not simply excessive worries about real-life problems. The person attempts to ignore or suppress them or to neutralize them with some other thought or action. And the person recognizes that these obsessions are a product of their own mind. Compulsions, on the other hand, are defined as repetitive behaviors, for example, hand washing, or mental acts, praying or counting, for example, that the person feels driven to perform in response to an obsession or according to rules that must be applied rigidly. Compulsions are aimed at preventing or reducing distress or preventing some dreaded event or situation. However, they're not connected in a realistic way with what they're designed to neutralize or are clearly excessive. So with the example I mentioned of contamination obsessions, certainly a expected compulsion would be hand washing, and that, of course, to some degree can be a normal response to a concern about contamination. But if hand washing is occurring to the point where it's taking up a great deal of one's time and one's hands are becoming red or raw, that's clearly excessive and can be associated with impairment, which leads me to the next aspect of the OCD phenotype. It's very important to point out that clinical OCD lies on the tip of an iceberg. So we know that there's a normal population variance in obsessive compulsive symptoms. And in the upper end of that, we have clinical OCD occurring in 1% to 2% of the population, at least in terms of lifetime prevalence. And we know that when one is in that 1% to 2%, that's because one has marked distress. The symptoms are time consuming, arbitrarily defined in the DSM-5 as taking up greater than an hour a day, or they significantly interfere with one's normal routine, occupational or academic functioning, or usual social activities or relationships. Like most DSM-5 diagnoses, one must demonstrate that the symptoms are not due to another axis 1 disorder and that they're not due to the direct physiological effects of a substance or a general medical condition. And there are two specifiers for OCD diagnosis, with or without insight, and tic-related or non-tic-related OCD. We know that certain disorders are commonly comorbid with OCD. So for example, there's a whole category known as OCD and related disorders in the DSM-5. In addition to OCD, this category includes hoarding disorder, hair pulling disorder or trichotillomania, body dysmorphic disorder, and skin picking or excoriation disorder. And these disorders are grouped together largely because there's a great deal of phenomenologic overlap and comorbidity, but there's also thought to be a potential biological relationship as well. That's certainly also true for another category of disorders, tic disorders, which commonly co-occur with OCD, and again are thought to have a biological relationship, and that's why we are studying these disorders together in the TSOCD working group. Mood and anxiety disorders are commonly comorbid with OCD, and in children and adolescents, we do see a high rate of ADHD as well. OCD begins early and is a risk for later mental illness. So about half of OCD begins before the age of 15, and it's thought that the onset may be bimodal, with an early onset form, mean age of about 10 to 11, and an adult form, mean age of onset of about age 21. And these two groups appear to have distinct features. So children who develop OCD tend to be male. There's comorbid tics quite commonly, and there's a positive family history, so important for our genetic studies. Long-term outcome studies of children who develop OCD indicate a 50 to 60% persistent after 10 years, depending on how persistence is defined. So there certainly are many people who go on to have OCD, but there are also many people who develop OCD in early life who go on to have other disorders, especially major depression and generalized anxiety disorder. So it's certainly a risk for the development of later mental illness in general, not just OCD. OCD is also a heritable neurobehavioral disorder. So we know that there's genetic causes for OCD based on twin and family studies, and that this heritability appears to be higher in children versus adults. It's a biological disorder, but it waxes and wanes in course, and that waxing and waning course appears to be potentially quite sensitive to environmental stressors. We know that OCD is associated with changes in corticostriatal thalamocortical circuits based on neuroimaging studies. And we also know from neurocognitive studies that there appears to be altered cognitive processing associated with OCD. So differences in things like cognitive flexibility and error processing in OCD individuals compared to controls. So I hope that that was helpful in giving you a very brief introduction to what is OCD. And I'd like to thank, of course, all of our patients and contributing sites, all the clinicians who make the lives of OCD sufferers better and also play an active role in contributing to our studies, all funders in individual countries, and thank you also for your attention.
Hello, my name is Jeremiah Scharf. I'm a clinician investigator and neuropsychiatric geneticist who specializes in genetic research of Tourette syndrome, OCD, and related disorders. In this segment, I hope to define the basic clinical aspects of Tourette syndrome and other tic spectrum disorders. So what are tics? Well, tics are rapid, arrhythmic, stereotype movements that most commonly involve the head, neck, and arms. They occur in repetitive bursts or bouts, and they tend to wax and wane over time. The key feature of tics that separates them from other types of movements is that they are usually partially suppressible. They're somewhere between voluntary and involuntary, and are preceded by a premonitory itch or urge or tension that is relieved when the individual allows the tics to happen. They're also clinically separated into different subtypes, motor and vocal, and simple and complex. From a classification perspective, we now think of tics as occurring along a continuous spectrum of developmental tic disorders. The most common transient tic disorder is classified by the presence of single or maybe multiple tics that are present for at least a few weeks, but disappear before 1 year and never return. This is now called provisional tic disorder in DSM-5 because you really can only make this diagnosis in retrospect. Of note, research suggests that approximately 25% of all school-age children will have transient tics. However, once tics have been present for at least a year, below the red line, we can see the diagnoses of a chronic or persistent tic disorder. Now, if an individual just has motor tics, they're given a diagnosis of chronic or persistent motor tic disorder. And if they only have vocal tics that last for a year, they have chronic or persistent vocal tic disorder. If someone has multiple motor tics, at least 2, and 1 vocal tic, which together have been present across the course of at least a year, they are given a diagnosis of Tourette syndrome. This is required to occur during development, so onset before age 18, but there now are allowed to be periods when no tics are present, often because patients may not recognize that they have tics for certain periods of time. We think this is now occurring along a continuous biological spectrum with no clear distinction between persistent tic disorder and TS. It's not yet clear whether transient tic disorder has the same underlying biology, but this is likely. I do want to make the point that patients do not have to have verbal tics involving words to have a diagnosis of TS. And in fact, any tic, sniffing, coughing, that makes a sound is considered a vocal tic. Now, why are we talking about a disorder of movement in a psychiatric genetics conference? This is because TS and other tic spectrum disorders are often associated with 1 or more developmental psychiatric disorders. In one recent study of 1,374 individuals with TS who were assessed with formal diagnostic instruments for DSM-4 psychiatric diagnoses in the context of a family genetics study, greater than 85% had at least 1 DSM-4-TR comorbid psychiatric disorder, and 58% had at least 2. Now, the most common were obsessive compulsive disorder, OCD, and ADHD, each present in approximately 50%. And 1/3 of individuals with Tourette syndrome have both OCD and ADHD. Now, while many other, essentially all other DSM-4 psychiatric disorders are increased in Tourette syndrome patients compared to the general population. The association with all these other disorders in a family genetics study were better explained by shared heritability with OCD or ADHD and not directly with TS. There is also strong evidence that the underlying neuropsychiatric dysfunction in TS can be attributed to a well-characterized brain circuit connecting the cerebral cortex to subcortical structures known as the basal ganglia, feeding back to the thalamus, and then returning to the same place in the cerebral cortex to give feedback. Now, this feedback is based on other internal or external input from other parts of the brain to help prioritize movements in the appropriate context. Now, these loops, known as corticostriatal thalamocortical circuits or CST circuits, also underlie associative cortex for learning and limbic cortex, such that it is now thought that there is shared common circuitry of these CSTC circuits and their dysfunction across TS, ADHD, and OCD. And thus, this may explain their shared and overlapping genetics. So to summarize, TS is a neuropsychiatric disorder defined by the presence of persistent motor and vocal tics. It's important to note that coprolalia or other complex tics are not required for a TS diagnosis. We now think that this is a continuous spectrum of developmental tic disorders, and TS is rarely an isolated tic disorder, with OCD and ADHD being the most common. This underlying abnormality in corticostriatal-thalamic circuitry may offer us an opportunity to link genetic susceptibility to biology. Please, if you have any other questions, I recommend that you go to this website or email either myself or my co-chair of the PGC TS-OCD working group, Manuel Mattheisen. Thank you very much.
Hello again. My name is Jeremiah Scharf, and I am the co-chair of the PGC TS-OCD working group. In this talk, I'd like to summarize our current knowledge about the heritability and genetic architecture of Tourette syndrome and related tic spectrum disorders. Tourette syndrome is one of the most highly heritable non-Mendelian neuropsychiatric disorders. First-degree relatives of individuals with TS have a 10 to 20-fold increased risk of TS compared to the baseline prevalence of 1% in the general population, and an additional increased risk of persistent tic disorder. Small twin studies suggested that TS was highly heritable. However, it was only in the last 5 years that large-scale population-based estimates of TS risk were conducted. 2 parallel studies using the Danish and Swedish national registries demonstrated a TSCT sibling recurrence risk is nearly 20-fold higher than that of the general population, while the increased risk to a child of a parent with TS or PTD is about 60-fold higher. These analyses also estimated a TS-PTD phenotypic heritability to be 0.77. However, despite 30 years of genetic research on TS, prior to 2017, there were no genome-wide significant TS risk genes identified in linkage and exome sequencing studies of large multi-generational TS pedigrees. One gene, HDC, encoding histidine decarboxylase, was identified as having a nonsense mutation in all affected members of a single family, and therefore is a strong TS candidate gene, though additional individuals harboring HDC mutations have yet to be identified. In 2017, 2 parallel studies were published identifying the first genome-wide significant loci for TS. Huang and colleagues identified 2 recurrent genic CNVs, a deletion in the 5' end of the neurodevelopmental gene, Norexin-1 alpha, and a duplication in another neurodevelopmental gene, Contactin-6. While the Norexin-1 deletions were the same proximal deletions found in individuals with various other neurodevelopmental disorders, and many of the Norexin-1 carriers in this study had both TS and ASD, none of the subjects with Contactin-6 duplications were known to have another neurodevelopmental disorder. For the second study, in parallel, an exome sequencing study of 777 TS parent proband trios and 1,153 control trios, Jeremy Wilsey and colleagues identified 2 genes with recurrent de novo likely gene damaging mutations, WWC1 and CLSR3, each with a Q value of 0.07. They also conducted de novo CNV analyses and estimated that approximately 10% of simplex TS cases harbor either a de novo deleterious CNV or a de novo likely gene damaging SNV, compared to a rate of 5% in the general population. Now, moving to common variation, this past year, the PGC TS Working Group published the second TS GWAS of 4,819 TS cases and 9,488 controls. The primary analysis identified one genome-wide significant SNP, known to be a cerebellar expression QTL in the gene FLT3. However, this locus did not replicate in the only available replication data set from Decode Genetics. In addition, the work group estimated the narrow sense heritability explained by SNPs tagged by GWAS SNPs using the linear mixed model package GCTA, and determined that the proportion of variance in liability explained by GWAS SNPs in the GWAS2 sample was 0.29. In comparison, a similar analysis in GWAS1 identified an H²G of 0.57, possibly because the first TS GWAS had a large proportion of familial cases, including 250 TS SIB pair families that might be more likely to harbor and be enriched for inherited common variation. Thus, to summarize, in this schematic, we estimate that from current data that rare variants may account for at least 5 to 10% of heritability, with common variants explaining somewhere between 30 to 60% of heritability, with known familial cases likely harboring a higher proportion of heritability attributable to common variation, and simplex cases being more likely to carry de novo gene-damaging rare variants. The last result from the TS GWAS2 study involved polygenic risk prediction using the TS GWAS2 sample for discovery and predicting case control status into the independent Icelandic sample of 706 individuals with TS, 466 individuals with non-TS tic spectrum disorders, and 127,000 population controls. Of note, in addition to predicting Icelandic TS case control status, TS GWAS2 polygenic risk could also predict case control status for non-TS tic disorders and demonstrated a significant, gradient of TS-PRS when comparing Icelandic TS cases to Icelandic non-TS cases, consistent with a model of shared polygenic risk across the tic spectrum, with the more severe phenotype being associated with higher TS polygenic risk. We also found, though not shown here, that in a familial TS cases, individuals with more severe TS, as measured by lifetime worst ever YGTSS tic severity scores, had higher TS polygenic risk compared to those with lower YGTSS scores. This leaves us with a theoretical model proposing a distribution of TS common variant risk across the entire general population. In this case, it is possible that individuals with slightly higher TS-PRS than the mean in the general population might represent the 20% of individuals who develop transient tics during childhood neurodevelopment, while individuals with chronic persistent tics may correspond with the 1 to 2% at the tail of the distribution, and those with severe persistent TS might represent those in the extreme 0.1% of the distribution with the highest polygenic risk. Of course, this model does not consider effects of other types of genetic variation, as well as environmental risk, but it may be useful to conceptualize the shared genetic susceptibility across the entire tic spectrum. Thank you, and we look forward to talking further in the open discussion portion of the work group meeting. Thank you.
Hello, my name is Manuel Mattheisen, and together with Jeremiah Scharf, I'm one of the co-chairs of the PGC TS-OCD working group. You have already heard about clinical aspects of OCD during this series of recordings, and with this presentation, we would like to provide you with an insight into the work we are doing in OCD genetics. If you have questions about the group or would like to contribute to its work, please consider to join us in our live sessions that will follow the recordings within the schedule of PGC Day, or contact us through our website at pgc-ts-ocd.info. The OCD half of the TS-OCD working group, similar to the entire group, is part of the PGC since 2013, and during this time has grown to include more than 100 investigators from numerous countries in Europe, North and South America, Asia, and Australia. Our aim is to study underlying molecular causes of OCD and related traits and disorders through large-scale genome-wide association studies. The data we are currently working with has been provided by more than 30 groups, in part as members of different national and international consortia, and we work with a broad variety of different sample types in order to provide us with the best opportunities to study different aspects of OCD. A large part of our efforts goes into genome-wide association studies of the categorical OCD phenotype. We have published the currently largest publicly available OCD GWAS in 2018 and are about to publish our next GWAS with 14,000 individuals with an OCD diagnosis and more than 500,000 controls on medRxiv soon. As part of this GWAS, we identified one genome-wide significant hit on chromosome 3, and through increase in sample size compared to our previous GWAS, could help to refine analysis into the genetic correlation with other psychiatric disorders, to name just a few. By the end of 2021, we plan to finish another round of GWAS analysis with a total sample size of up to 60,000 cases, which will allow us to delve deeper into downstream analysis of causal effects, functional annotations, and other analysis that will help us to understand the genetic basis of OCD. We are also working towards our first GWAS in samples of non-European ancestry and plan to integrate existing data from our own cohorts together with samples that are currently genotyped. As part of these efforts, we are also working closely with our colleagues in the IC-MAGIC consortium in India and other groups around the globe. During the last year, we have also established new subgroups to look into additional aspects of OCD. The first of these subgroups was the dimensional phenotype subgroup. The group is led by Paul Arnold, Dick Schmidt, and Sandra Meyer, and is currently conducting meta-analysis for both hoarding and OC symptoms after individual sample GWASs have been conducted in samples like the Netherlands Twin Register, the Swedish Twin Register, Twins UK, and Kits for Science. Amongst the different instruments used for the OC symptom studies are the Obsessive Compulsive Inventory, the Padua Inventory, and the Toronto Obsessive Compulsive Scale. After a first round of analysis in population-based samples like the ones mentioned above, we are also planning to work with our groups in Australia and the UK to use samples ascertained to study depression and depressive symptoms to shed light on the overlap between OCD and depression. Another area of interest are the genetic factors that influence treatment response in OCD. Our members have individually already published extensively on the topic, and we would like to use the power of our collaborative work in the group to hopefully further our understanding of OCD treatment response. The group is currently in its infancy and provisionally led by David Matishkols, Pino Alonso, Gwyneth Lai, and Erica Normi until the full group has formed. Together, they currently identify suitable samples, of which they already found a few, and would welcome your contribution. The group will focus on all aspects of treatment response in relationship to genetic factors that might influence them. This includes response to pharmacotreatments as well as response to psychotherapy. Following in the footsteps of and in close collaboration with our MDD counterpart, that is our treatment response working group, we hope to add to the picture of treatment response studies in pharmacogenetics and CBT. Last but not least, we are also about to establish a copy number variation subgroup. The group will be provisionally led by Jim Crowley, and we are still looking for members to co-chair the group with him. So far, smaller study samples, in contrast, for example, to the recent schizophrenia studies, have been studied, and their size hindered a more detailed insight into the effect of copy number variants on OCD risk. We hope to change this through a collaborative effort in this subgroup. So if you're interested in our work, want to know more about us, or become a member and/or have samples to contribute, please consider to stick around until the end of these recordings and join us during our live sessions during PGC Day. Alternatively, please visit our website at pgc-ts-ocd.info and get in contact with us. With that, I would like to thank all of our participations and our contributing sites, all the union and not so union investigators and clinicians that spent numerous hours to make our work a success. And I would like to thank you for your attention.
Hi, this is Jeremiah Scharf again on behalf of the PGC TS-OCD Working Group. Here, I would like to describe briefly the aspects of TS and tic spectrum disorder genetics that are currently under study by the PGC. As we noted in the annual general meeting, last year we completed the second TS genome-wide association study consisting of 4,819 TS cases and 9,488 controls. We did identify one genome-wide significant SNP in the FLT3 locus, but as I mentioned in the last segment, we were not able to replicate this finding in an independent sample. However, we were able to obtain a multi-center NIH grant to conduct the next wave of TS genetics and are currently analyzing our TS GWAS3 study of 13,000 TS cases and 50,000 controls, with expected preliminary results by the end of the year and more comprehensive analyses sometime in 2021. In parallel, the TSCNV group is currently assembling samples and an updated analytic pipeline for the follow-up to our 2017 TSCNV2 analysis. At present, we are expecting about 8,000 TS cases and approximately 20,000 controls that would certainly welcome any additional samples for either the TS or CNV analyses from current or new members of the group. A third area of interest of the TS subgroup is analysis of alternative TS phenotypes that might correspond more closely to the underlying biology of the disorder than the more heterogeneous categorical phenotype. One example of an alternative tic phenotype is represented by this item-level factor analysis of 1,200 individuals with TS and their 2,300 first-degree relatives derived from the tic symptom checklist of the Yale Global Tic Severity Scale. The best-fitting five-factor model paralleled the rostral-caudal or head-to-toe progression of tic onset, as well as both simple and complex tics that also may correspond to the somatotopic organization of body representation in the sensory motor portions of the corticostriatal thalamocortical circuitry thought to be dysregulated in TS pathophysiology. One can see on the left how the primate basal ganglia is parceled into simple motor movements arising from primary cortex, M1 in red, and projecting to the lateral putamen, while more complex motor movements arise from the supplementary motor cortex in green, and also project in a somatotopic fashion to more medial regions of post-commissural putamen. This circuit mapping may provide an anatomical correlation to one aspect of the significant variation in tic severity that is observed clinically in this disorder, and also might have an underlying genetic component. Lastly, I want to mention two brief examples of using TS polygenic risk scores derived from PGC TS GWAS studies to probe the relationship between TS and other heritable disorders, as well as to try to localize the brain regions and cell types most enriched for TS susceptibility variants. Here is an example from the Brainstorm Consortium, published in 2018, representing an analysis using GWAS summary statistics from 10 PGC disorders, as well as those from 12 neurological disorders, to examine the degree of genetic correlation across these 22 brain diseases. While there were multiple notable findings from this study, I wanted to point out one specific to TS, in which the cross-disorder analyses identified an unexpected shared genetic relationship between TS and migraine that had never been considered to be biologically connected prior to this study. And finally, the three primary analysts from the TS GWAS2 study we're able to use the cumulative summary statistics from this study to probe expression QTL datasets from the ENCODE consortium to identify specific human tissues with EQTLs most enriched for aggregated TS polygenic risk. While the only statistically significant result was in dorsolateral prefrontal cortex, 9 of the top 10 tissues represented nodes within regulatory CST circuitry, as well as cerebellum. We look forward to the ability to extend these analyses in the upcoming TS GWAS-3, and also to integrate information from new types of non-coding regulatory regions, such as promoters and splicing regulators, as well as single cell data sets that have the opportunity to identify specific cell types most enriched for TS polygenic risk. Thank you. And again, we look forward to talking further in the open discussion portion of the work group meeting.
Hi, my name is Christie Burton, and I'm the Social Outreach Liaison representing OCD. And I'm here to talk to you about what social outreach is, why we do it, what we've been up to, and how you can get involved. So social outreach has been part of the TS OCD group for about a year, but it's part of a broader PGC social outreach effort. And the aims of that effort are to help PGC scientists or members engage with other scientists as well as the public, and to help translate findings from the PGC, but about psychiatric genetics more broadly, to the public. And we're hoping to engage with people primarily through the website, but also through social media. And the idea is for the PGC to become the trusted source of information of all things psychiatric genetic. Now, for our own working group, I see this as a really good opportunity for us to promote the work that we do and give us a chance to help educate the public about the genetics of TS and OCD. So over the last year, we've been working to update the PGC main site, which is undergoing a reorganization. So we've updated the TS-OCD content on the main page, and that's active now. And then we've been working on creating some new content that will become active when the new PGC site is launched. So this includes a for the public section that will have a section that explains some important concepts in psychiatric genetics, but also a whole bunch of resources for each disorder, which includes TS and OCD. And so I wanted to say a special thank you to the working group members that helped me generate that list of resources, and those should hopefully be available on the website soon. We've also been creating our own TS-OCD website, and I'll give you a little glimpse of that in a moment. And finally, for those of you who don't know, the PGC has a Twitter account. The handle is @PGCgenetics, so give it a follow if you use Twitter. It's been relatively inactive over the last year as we've been reassessing our social media strategy. So over the upcoming year, we're hoping to launch the new PGC website as well as finish our TS-OCD site. And the links for both of those are below. We're also hoping to update the TS genetics and OCD genetics content on Wikipedia. So it's actually not too bad compared to some of the other disorders, but we'd like to update it nonetheless. We're also relaunching the PGC Twitter account. So the new plan is for each working group to have a 2-week period where they take over PGC Twitter and share everything to do with their disorder. So we're first up to bat March 28th to April 11th next year, and I will definitely be reminding you about that before it happens. And we're also hoping to create some short videos for each working group that can go on the website to help educate the public about concepts related to OCD and TS genetics. So if you go to our website, and the link is below, and you haven't been before, you'll see this little pop-up that lets you know that the site is under construction. It also gives you the opportunity to enter your email if you want to get updates from the group. And this is what the website is going to look like generally. So this is actually a picture of our working group from last year at World Congress when we could actually see each other in person. So we're still very much adding content to the website. If you're interested in creating content for the website, or you have any ideas about bits and pieces that we can add or change, definitely please get in contact. We are all ears. And there's generally a lot of ways for you to get involved if you're interested. So once the website is live, we're going to need to update it with new publications that use the PGC data, new working groups, anything new and exciting relating to the group we can add to the website. And I'll be bugging you about those types of updates quarterly by email. We also would love to get content for the PGC Twitter feed, so ideas for tweets, studies, resources, anything that you want to share related to TS and OCD. It does not have to be specific to the PGC at all. And generally, it's a really good opportunity to share work that you and your group is doing. It also doesn't have to be in a social media friendly form. I just need an idea and I can do the rest of it myself. But if you are interested in creating content for the Twitter account, also please let me know. We're also looking for individuals with experience uploading content to Wikipedia successfully. So if you happen to know anybody, please get them in touch with me, please. And also, we're just generally interested in any new ideas related to outreach. So if you want to get involved in any way, shape, or form, please reach out to myself if you're in the OCD group, or to Perry, who is our social outreach liaison for TS. We'd both be happy to chat with you. So that's it for me. Look forward to hearing from you. Thanks.